lot of chapter 2 starts to talk about light and the properties of light, and I'm going to link you guys to a video that'll help you understand that a little bit better from a physics perspective. What I'm going to primarily focus on here in this video is the relevance of that light, which comes back to microscopes, and so we're going to talk primarily about microscopes. So one of the first things that you want to do in relation to this video is I tell you to make sure that you label the parts of a microscope and the function of each one of those parts. And so this picture is straight out of the textbook, but I have added, and now I have just realized I need to swap those two views, sorry. I have added the function of each part if it didn't already mention it in the description there. So to run you through things. Number one up here is pointing at the oculars or the eyepieces. In our lab, the oculars magnify by a factor of 10. They're adjustable, so you can rotate them in and out. When you look through those eyepieces, you just want to see one visual field. If you're seeing two, you really want to adjust those so you don't end up with a headache. Number two is pointing to the nose piece. It's this little black ring that holds the objective lenses. You can rotate that around to switch from one objective lens to another objective lens. Number three is the objective lenses. These are what I usually call the powers. There's 4 different powers that are on our microscopes. We've got scanning, which is red, low power, which is yellow, high power or high dry, which is usually blue, and then the oil immersion lens, which is either white or black, depending on which microscope you're looking at. Our microscopes are designed so that you don't have to lower the stage to rotate things around as long as you have the slide on correctly. And so once you've found it on a lower power, all you have to do is rotate the nose piece around to the next highest power, and then you'll use the fine focus to bring it back into focus. So the objective lenses are those powers. Number 4 is off over here. That's pointing at the coarse focus knob. You're only going to use the coarse focus knob when you're on the scanning objective, because you really just use that one so that you can actually find the object that you're trying to see. You don't really bring it into crisp, clear focus with that one. That's the job of number 5, the fine focus knob, which most microscopes have 2 of those, one on either side. The fine focus knob is what you'll use when you get up to the higher powers. If we catch you using coarse focus when you're on a higher power, we're going to knock you on that, because you can actually break the slide if you use the coarse focus knob when you're on those higher power objectives. So make sure you're only using fine focus when you get to those higher powers. Number 6 is pointing at the stage. You put your slide on it. Now, a lot of times in high school, they have, for lack of a better term, crappy microscopes that have these little clips, and you're supposed to put the clips over the slide. That's not how our microscopes work. There's an arm. You put the slide up against the corner of the mechanical stage, and then you just let the arm go, and it's going to rest against the corner. It's not going to go over the slide, though. If you put it over the slide, your mechanical stage is not going to work correctly, so you just let the arm rest against the corner. Number 7, your illuminator is just your light source. The only note that I really have for you guys there is it can get really hot. 2 ramifications for that. 1, don't touch it. You can burn yourself. But 2, the longer you leave a slide on your microscope, the more likely you are to dry out your slide if you're doing a wet mount, or flat out kill the organisms that are on there, because that light is just so hot coming from down the bottom. Number 8, there's 2 little parts that are going on down here, and it's kind of hard to see which is which as you're looking at them, but the condenser is this structure that's underneath the stage. It's taking the light and it's condensing it to this small little circle that's going to come up from the top. So this little ring right here is also part of the condenser. The condenser, you can move it up or down. I tend to like to put it right under the slide, but it does depend on what you're viewing. So you can play around with the condenser. There's usually either a knob or there's a lever that moves up or down. It just depends on which microscope you have. Now, towards the front of the condenser, there's a diaphragm. This little lever you can pull right to left, and it controls how much light is passing through the condenser lens and actually hitting the object that you're trying to view. One of the things that our students tend to do at first is they want everything as bright as possible. They've got the light turned all the way up. They have the condenser as bright as it can be. The diaphragm is all the way open, and that can really hurt your eyes, fry your eyes as you're looking at things for a long period of time. So play around with these and adjust them so that you can see things as best as you possibly can without frying your eyes at the same time. It'll really give you a headache if you've got it on bright the whole entire time. So there's that. Number 9, the mechanical stage itself. This is the apparatus that holds the slide and then it attaches to these two knobs, which this is calling them the XY mechanical stage knobs. I usually just call them the stage adjustment knobs, but whatevs. What this will do is one of them is going to move the slide back and forth across the stage, right and left. The other one is going to move the slide back and forth, up and down across the stage. If you're doing things like you're counting how many basophils are present on a slide, you will rhythmically and in a very regimented fashion move your slide all the way across right to left, move it down one click basically, and then move it all the way in the other direction so you can count all of the basophils that are on that slide. If you've got something that's swimming around and is trying to swim out of your field of vision, you'll use these knobs to follow it around. One of the important things to keep in mind there, though, is that because of the lenses that are in here, everything that you view in a microscope is upside down and backwards. So if your object looks like it's swimming off to the right, it's really swimming off to the left. So you tend to have to move the stage in the opposite direction of what you think you're going to have to move it. Those are the only things that have numbers. So let's see what they've got up here that doesn't have a number. I can't even really. Okay, so this is trying to point at those two knobs that I was just describing what they do. The rheostat is in different places on different scopes, but this just controls how bright and dim the light itself is. And again, I'm going to recommend you don't start with it at maximum brightness just because it really will fry your eyes over time. I think that looks like everything that was on here that didn't have a number associated with it because I already hit both of those. So those are the parts of a scope. Now you're going to get all of those again in lab, but I mean, it's microbiology. You can't do microbiology without a microscope. So you got to make sure you know the parts and you know how to use it. Most of the students that are coming out of freshman bio have a pretty good idea how to use a microscope, but students that are coming out of A&P, everything was already set up for you and you didn't have to do anything. And so you guys are kind of have to play catch up as you come in here and try to learn those parts of a microscope. Other things that are kind of relevant, when you first start to view objects under a microscope, they have very low contrast for the most part. Now, there are some organisms that have these structures that are called chromophores in them. They are structures that have a pigment to them, and that pigment is going to absorb certain wavelengths of light and then reflect others. The best example that I can give you for that coming out of high school, this is an Alodia leaf under the microscope. And these little green structures that you can see, well, that you could see a second ago, it'll come back into focus in a second. Those are chloroplasts. Chloroplasts have chromophores that are made out of chlorophyll, which is the pigment that actually makes them green. So those you don't have to stain, they just look that way. Also, just as a PS, what you're seeing in this video is something called plasmolysis. We added some salt water to this slide so that you could watch the plasma membrane shrink away from the cell wall. But anyway, you can still see all the little chloroplasts because they have pigment. Most bacteria don't. Some do, but most do not. When you guys viewed cheek smear slides in whatever class you had prior to this, you had to stain it. The stain adds chromophores in that case. Okay, another thing that comes in. Usually in A&P, you do not get to use the oil immersion lens. And very often in freshman bio, they don't want people using the oil immersion lens because it's a lens that takes a lot more maintenance than a traditional objective lens is going to. The oil immersion lens on our microscope magnifies by a factor of 100. And because it is such a high magnification lens, it tends to have low resolution if you're passing the light just through the slide and then to the lens. So much of the light gets bounced out, like what it's trying to show you here in this picture. So everything ends up looking very blurry as you look through the oil immersion lens if the oil isn't present. So what you do is, as you're moving from high dry to the oil immersion lens, you put 1 drop of oil on the slide that you're trying to view, and then you move the oil immersion lens so that the lens is actually going to dip into the oil that you put on the slide. Now our light is not passing from glass through air to the lens, it's passing from glass through the oil, which has a very similar refractive index to the glass, and so we're not going to lose as much light. It's going to focus the light that much better on the lens so that you're going to be able to see better. So this improves resolution. Now, students tend to be scared of using the oil immersion lens at first because, again, it does take a lot more maintenance. You have to clean it every single time after you use it, and cleaning it is a pain in the butt. But if you're ever sitting there and you're going, I'm not really sure if this is a coccus or bacillus, it's so small, take it up to the oil immersion lens and you'll be able to tell exactly what it is. So don't be afraid to use it in micro. You need to a lot of times when you're in micro. Okay. One of the things that you guys are going to do in lab is create a wet mount. The benefit of doing a wet mount is that you actually get to view living organisms on the slide. One of the first ones that we usually do in micro is we have you guys view yogurt under the microscope. And yeah, yogurt has milk solids in it, but it also has those live and active cultures that it talks about on the label of the yogurt. That's the bacteria that's going to be actually swimming around within the medium. And so you'll be able to see the bacteria using their flagella and swimming around on that slide. Now, this is actually Vorticella. It's a protist as opposed to bacteria, but these little bitty specks that are moving around, those are bacterial cells, and the Vorticella is actually creating a current to eat those. You wouldn't be able to see those if you created a heat-fixed emulsion and then you stained them, because that would kill the Vorticella. By doing a wet mount, though, you can actually see them actively feeding, and so that's one of the benefits of doing a wet mount. One of the procedures that you're going to spend a lot of time doing in lab is the gram stain. It's very regimented again in how you have to do it. There are 4 steps. They all have specific times that you have to carry out. If you don't do them long enough, you're not going to stain things correctly. And if you do them too long, you're going to get false results as well. And so this is one of those things you're going to practice time and time again when you're in the lab until you get it right. But it's one of our types of differential stains. What it does is it stains some organisms purple because of the crystal violet, and then it stains other organisms pink because the crystal violet gets washed out and we counterstain with safranin. The ones that stay purple with the crystal violet, those are called gram-positive organisms, like these coccus-shaped organisms that are on this slide. The ones that get the crystal violet removed and then counterstain with safranin, those end up being pink, and those are gram-negative organisms. So the bacilli on this slide are the gram-negative organisms. This is, again, one of those things that takes a lot of finesse as you're doing it in lab, and it is very easy to get both false positive and false negative results if you don't do the prep exactly correctly. So there are 2 mistakes that we tend to see the most often in the lab. First one is the student is so concerned about being able to see their organism on the slide that they put way too much organism on the slide. When you do that, you end up with these huge clusters of organisms like in this pile right here. And then when you try to decolorize that, since you're only decolorizing for 10 to 15 seconds, it would decolorize these really easily, but it's going to have trouble getting into this large pile of organisms that are in this overly smeared part of the slide. So this is going to give you a false positive result because the decolorizer, the alcohol, didn't actually have time to decolorize this. So don't put too much organism on there. You want just enough so that you're going to be able to decolorize correctly. The other thing is, now decolorizer, all the other steps are really easy. You put them on, you leave them on for exactly a minute, and then you rinse them off. The decolorizer, though, is one of those things that you leave it on for a different amount of time depending on how thick you put your emulsion on there. You tend to want to leave it on just for 10 to 15 seconds. If you go longer than that, you will decolorize even the gram-positive cells and end up with a false negative result. Don't decolorize long enough, and then everything's going to look gram-positive and you get a false positive. And so you really have to find that fine balance between decolorizing too long and decolorizing not enough, essentially. We will usually give you fresh samples, but it is important to know that if you use an older sample, it will tend to give you a false negative result. And so that's something that we'll just prepare for you. Okay, last thing I wanted to mention in this presentation has to do with the clinical focus that goes throughout the chapter. It talks about a girl that ended up with a little infection. The infection that she had is with a bacterial species called Staphylococcus aureus. Now here's one of the beautiful things about the name. The name means something. Coccus goes back to the shape, which the organism is coccus shaped. That's the answer to one of your questions. Staphylo is a prefix that means they cluster together. And so as you look at this, it looks sort of like clusters of grapes. Well, that's what Staphylococcus means. It means cluster of ball-shaped cells. Aureus actually means golden, and that's because when you let these grow on an agar plate, they make a golden sort of yellowy colony, and so they look golden. All right, the gram reaction, you should be able to tell from this picture. They are purple. Purple meant something. What did it mean? Purple meant positive, so they are gram positive. Okay, from there, MRSA. So ordinarily, if you get a staphylococcal infection, If you aren't allergic to it, we can just treat you with penicillin and it's going to take care of the infection. If penicillin is contraindicated for some reason, we can treat you with methicillin unless you're allergic to that. We're not going to talk about allergies though for the most part right now. If you have a MRSA infection, that's a specific strain of Staphylococcus aureus that is resistant to both penicillin and methicillin, which means we cannot use those two antibiotics to treat your particular type of infection. The standard treatment for that in the case of MRSA is an antibiotic called vancomycin. Now, vancomycin is not something you can go and get a prescription for and take orally for the most part. You have to get IV infusions of vancomycin. It also happens to be way more toxic than penicillin and methicillin, so vancomycin is contraindicated for some people, especially diabetics with kidney disease. What that means is for people that have MRSA, it is more difficult for us to treat them. Now, here's another problem that really isn't discussed in this clinical focus thing. Since vancomycin is the go-to treatment for that, we need vancomycin to work basically all the time. It's our last resort for staph infections, and we are already seeing strains of Staphylococcus aureus that are what are known as intermediate and what is known as resistant. So let's start with Visa. Vancomycin intermediate means you could use vancomycin to treat it, but you're going to have to do higher doses for longer periods of time, which means we're going to see way more side effects associated with that. Versa is vancomycin resistant, which means vancomycin doesn't even work for that one anymore. So one of the reasons that we're seeing a rise in antibiotic resistant is people tend to stop taking their antibiotic as soon as they feel better. And then they'll do what I'm going to call save for later. So they'll take like half of their antibiotics and they'll save the other half for some later infection that they see themselves having at some point in the future. The problem with that is when a patient or you do that, you didn't actually kill all of the organisms that were causing your infection. Because here's the thing, you aren't just infected with one strain of a bacterium. There are very often cells that are different within your infection. If you happen to have an infection made of a bunch of different strains of Staph aureus, some of those strains are probably going to be resistant to the antibiotic. And if you stop taking it early, you did not kill those cells. Then when you stop taking it, those cells are going to start to reproduce and you're going to come back with a flare-up of your infection. And that flare, and I just realized I have a typo on my slide. Anyway, I'll fix that later, I guess. I get distracted so easily, sorry. But what it means is now you can't use that same antibiotic to treat the infection. You're going to have to go for an antibiotic that is more potent and is probably going to have more side effects. So if a doctor gives you a prescription for something and they say you need to take this twice a day for 10 days, you need to take it for all 10 days. If they give you something and say you only have to take this for 3 days, you have to take it for all 3 of those days. Follow the instructions that you're given, because if you don't, you are more likely to end up getting a resistant infection in the future because you didn't kill them all the first time around. It's also very important that patients get educated about that. Make sure that all patients know if they have been prescribed antibiotics that it is very important that they take the entire prescription of those antibiotics. And that's not just for people. If you have pets, dogs or cats, and they get an infection and they get antibiotics, they need to take the entire round of antibiotics so that they don't get resistant infections, because sometimes infections in animals can spread to people. And so we just really want to try to prevent that in all of its nice little iterations. Hopefully that helped get you through the more difficult things in this chapter. The rest of this chapter is really fairly straightforward except for the light stuff, but again, I'll give you a different video for light. Hope this helps.